Hello students, in this part we will discuss on linkage and recombinant. Linkage and recombinant. What is linkage and recombinant? Recombinant means when one pair or two pairs of parental traits are crossed with each other, the progeny which is produced, that is non-parental, means that is not similar to their parent, that is form a new generation, new types of progeny is known as recombinant. Link is physical association of gene on same chromosome. Physical association of gene on the chromosome, that is known as linkage. How this is required, there is need of linkage and recombinant. After the Mendel's dihybrid growth, Mendel assured that genes control the characters and they segregate from each other independently and show the definite ratio. That was not surety. Thomas Hunt Morgan showed the genes may be separate individually from each other when they are present on different chromosomes. But the location of genes on the chromosomes are similar, same chromosome as well as less. Then there is no surety to segregate the genes from each other. We will discuss on this part. After the Mendel's dihybrid growth, he assured that genes control the characters. Genes control the characters and segregate from each other independently and oscillate a law of independent assortment. Law of independent assortment. It means when genes are crossed with each other, genes separated segregate from each other independently. He saw in the Mendel showed this one, but there is no surety because we know that genes are present on chromosome linear, means linear present होते हैं. Suppose this is the chromosomes. Genes A, B, C and D. If genes are present here, not other genes. These genes are present in the other side, other chromatin this side. No, genes always present in linear, means E, F. But the segregation may be not sure every time genes may be segregated. This depends on the gene's location on chromosomes. We know that genes are present on chromosome linear, linear. Genes present on chromosome linear, but there is no surety every time crossing process may segregate the genes. Suppose these are chromosomes. If one gene is present here and other gene is present here, then during the crossing process, the genes may be segregated. There is chance of genes may be segregated because crossing process means non-sister chromatin या sister chromatin होते हैं एक दूसरे से vibrant बनाते हैं non-sister chromatin, then they segregate. This is the chance of segregation. If the location of genes on chromosomes is closely, suppose one gene is present here and second gene is present here, and during the crossing process there is no chance of segregation because there is no genes present. So after the law of independent assortment, Mendel showed that every time genes segregate from each other. This is not surety. Thomas Hunt Morgan. Thomas Hunt Morgan. Experiment on Drosophila melanogaster. He experimented on fruit fly. He experimented many experiments on fruit fly. Then he saw that not every time the genes segregate from each other. It depends on the location of the gene on chromosomes. So there is no chance of segregation. He showed this, how the genes are not segregate or segregate from each other with the help of experiment. How he showed? He experimented on white body and yellow body. He took yellow body, white eyes, female. He crossed yellow body, white eyes, female with brown body, red eyes, male. He crossed yellow body, white eyes, female Drosophila with the brown body, red eyes, male Drosophila melanogaster. Female. In Drosophila melanogaster, the chromosomes may be similar, homochromosomes. This is the homochromosomes. X and X. X chromosome in male, and in female, XX, and in male, one chromosome is X and other one is Y. This one is X, this one Y. He crossed yellow body, white eyes, female with a brown body, red eyes, male. X chromosome show the position of the gene's location. Suppose yellow body represented by the capital Y and white eyes represented by W. So it all contains yellow body, white eyes, female. And the brown body represented by Y plus and red eyes with W plus. There is no genes present on Y, it present only XX. He crossed with each other in F1 generation. In F1 generation, he saw that all the female are brown body, red eyes, whereas male, yellow body, white eyes. How? Suppose X chromosome crossing with the X chromosome of male, then the chance of one is X chromosome of parental, this one, Y and W, and one X from the brown body, red eyes, male. X का इसका क्या था? Y plus and W plus. Y plus and W plus. So brown body, red eyes dominant, whereas yellow body, white eyes recessive. In one chromosome, it shows the parental, this one female and one from male. One from female and one from male. So the male dominant genes will cover this recessive. So all the females, this is the female because both are the X chromosome. This female, how the female is produced? Yeah, which type of body and eye color is in female? Brown body, red eyes, because one chromosome contains Y plus W plus, and Y plus W plus represent the brown body, red eyes. So in F1 generation, all females are produced brown body, and red eyes. यानी जो मेल का कलर था वो तो फीमेल में, फीमेल का जो कलर था वो मेल में चला गया। क्यों? इस एक्स का जितनी बार भी इस एक्स का इससे एक्स से कराया तो भी आपका यही रेशो आएगा, इस एक्स का दोबारा इस एक्स से कराइए तो यही आएगा। लेकिन इस एक्स का जब वाई से क्रॉसिंग कराओगे तो वाई देयर इज नो चेंज, वाई विल रिप्रेजेंट द एक्स क्रोमोसोम्स ट्रेट्स, तो देन वन क्रॉसिंग विद एक्स एंड वाई, it present one chromosome from here YW and this is the Y chromosomes, so it is X and this one is Y. X chromosome represent the gene, which one? Y and W. Y and W show the yellow body, white eyes. After crossing with each other in F1 generation, he saw that all the females are brown body, red eyes, whereas male yellow body, white eyes are produced. After the crossing or recombinant process, then all chromosomes, suppose this is the X, this one it makes its copy, this X makes its copy, it makes the YW, it makes its copy, then this one is also YW, means yellow body, white eyes, and this X is also make this copy. You can represent it with a different color. Y plus W plus. Again, during replication, it makes this copy. This one is also, we have take one pair of chromosome. It make its copy double. It also make its copy double. Y plus W plus. The crossing process between the two non-sister chromatin. Then the crossing process after this one, replication take place. Then after this one, crossing process can take place. Crossing process can take place. How is the crossing process? First one is the, we can take it from here. First one is the, this one like, it contains YW and this one is also YW, but non-sister chromatin, when the crossing process can take place, overlapping, Y plus comes to here and Y comes here. So exchange of genes can also take place. So it makes Y plus W, and the other one is यहाँ क्या क्या क्रॉसिंग प्रोसेस की, Y प्लस इसका इसके ऊपर ट्रांसफर हो गया, Y यहाँ आ गया, इसमें क्या रह गया Y प्लस W और इसमें क्या हो गया YW प्लस and it remains the similar. It remains the similar. Y plus W plus. So after the crossing process, again listen. Morgan crossed with Drosophila melanogaster, yellow body, white eyes, female with brown body, red eyes, male. In F1 generation, he saw that all the females are brown body, red eyes, whereas males are yellow body, white eyes. He again, suppose here is the recombination, replication process can take place, so all the genes, all the chromosomes make its copy. XY, YW, YW. It's make its copy. This one is also YW. This one is Y plus W plus, Y plus W plus. Then the non-sister chromatin crossing process can take place. Suppose this is the X, same YW. It mix. Y plus is exchange here and Y here. So it contains Y plus W and it YW plus. The last one is the similar. Clear? अब यहाँ पर ये क्या जनरेशन आ गई, ये जो पेरेंटल प्लांट आ गई, पेरेंटल नॉट पेरेंटल प्लांट, पेरेंटल फ्रूट फ्लाई, जो इंटरस्पीसीज में F1 जनरेशन के अंदर जो प्रोड्यूस होते हैं उनके कुछ इस प्रकार से कॉपी पॉसिबिलिटी ये आ गई। मेल क्या है, मेल तो एज इट इज है, मेल का तो कुल एज इट इज है, Y W, and this one is the Y chromosome. YW stand for yellow body, white eyes. This is the X and this one is Y. All these chromosomes firstly combined with X. Firstly, house the makes the female body. Then after that, we crossing process in F2 generation. F2 generation. Firstly, we will cross with all X with this X1. This X1 is YW with this one is also YW. YW both are similar. So the females which are produced is yellow body, white eye. If we cross with first X, this one, now crossing with this X with this X1, then the second condition, one is Y plus W, because this is the crossing process. Y plus W cross with YW. YW. So one it contains Y plus. Y plus show for the, suppose yellow body is the Y and white eye, color of eye stand for white W. Here brown body stand for Y plus and red eye stand for, any red eye stand for W plus. Here is a Y plus. It means the body of the Drosophila in this one female is brown, but the eye remains white. Brown body, white eyes. This is the separate new generation which is not present in parental. Parental both are separate. Brown body, red eyes. It contains yellow body, white eyes. This one brown body, white eyes. New generation which not resemble to its parental. So we can say that it is white. Clear? One pair of X cross with this X1. Again this X with this X1. Firstly we cross this one, then this one, and now this one. YW plus with YW. YW plus with YW. YW stand for yellow body, white eyes, and Y stand for, the Y is the yellow body, but W plus stand for the red eyes. So we can see that yellow body, because both have Y. Y stand for yellow body, and W plus and W plus is dominant, so red eyes. These are also separate from the parental, so are the non-parental. This one is also white. Same again, this Y plus W plus again cross with this chromosome. Again, fourth generation, Y plus W plus stand for, this one is the Y plus W plus, and next X is this one, YW. Here is the female produced which contain Y plus W plus. Y is dominant, Y plus is dominant. W plus and W, out of both, W plus is dominant. Y plus W plus show for the brown body, red eyes. Brown body, red eyes. So in female, there is, these are the possibility. Yellow body, white eyes, brown body, red eyes means wild, not parental. Yellow body, red eyes, not parental, wild. And this one is brown body, red eyes. These are the same like as parental. In female terms, F2 generation, all females are produced these one lifestyle. And again, all 4 X chromosome again reacts with Y chromosome. F2 generation में जो male produce होंगे, what are the possibility of F2 generation males? Crossing process can take place. This first X with the Y, X and Y. X and Y कौन सा यहाँ पर? Y and W. YW stand for yellow body, white eyes. तो male जो produce हो कैसा? Yellow body, white eyes. One male will produce this one line, and again this X with the Y. this x is y plus w, and y there is no genes present. So this male is, y plus stand for brown body and w stand for white eyes. So this male is brown body, white eyes. Also we can say that not just like parental, both property are not parental. Brown body with red eyes होनी चाहिए, या white eye with yellow body is not matched to the parental, so non-parental we can say that white. Second, third, YW plus with Y. YW plus with Y chromosome. It means this is the male, yellow body with red eyes. Yellow body with red eyes. This is not parental. Yellow body white eyes होनी चाहिए या brown body red eyes होनी चाहिए. Dissimilar, so we can say that white. And last one is Y plus W plus. Y plus W plus, and this one is Y. This one. So this male is Y plus W plus, show the brown body red eyes. Brown body red eyes. After crossing this process, he showed that every time there is no possibility of segregation of genes, because genes are present on chromosome. If the genes are tightly packed, then there is less chance of segregation, and genes are present at long distance, then there is more chance to segregate. He crossed, Thomas Hunt Morgan crossed fruit fly, yellow body, white eye with brown body, red eyes. After the second generation, F2 generation, he saw that 98.7% new progeny are resemble to parental. 98.7% progeny resemble to parent, but 1.3% progeny are recombinant, non-parental. Means 1.3% chance जो है उसके हैं non-parental के, जो form the parental से match नहीं करती, non-parental, which is known as recombinant. So on this basis, he showed that physical association of gene on chromosome, that is known as linkage. The linkage means physical association of gene on chromosome. The position of gene on chromosome may be segregate or not. If the distance between the two genes is higher, then there is higher chance of segregation. And if the genes are located tightly with each other, then there is less chance. He again crossed yellow body, miniature wing female with brown body, normal wing male. Same crossing process can take place. Here is the percentage ratio is different. 68.2% offspring a progeny resemble to parents, whereas 37.8% progeny are non-parental. Means इसमें recombinant कितने प्रोड्यूस होगी? Recombinant 37.8%. So in this crossing process the ratio is different. Parental Age जो progeny 98.7, non-parental means recombinant 1.3, और इसके अंदर क्या हुआ? Parental जो है 68.2, whereas recombinant 37.8. After this experiment, he showed that the Mendel proposed that genes are segregated every time, and the ratio 9:3:3:1 we cannot obtain every time. This depends on the position of gene on chromosomes. But how the genes presence on chromosomes? His student, after the Morgan experiment, Morgan's student Alfred Sturtevant showed that this different possibility of recombinant is present due to the position of gene on chromosome. He showed that if the genes are tightly packed, then there is less variation can take place, and if the genes are loosely packed, they have large distance, then the chance of variation may be higher. How is the distance measured between the genes on chromosome? He postulates the unit that for the measurement of the genes on chromosome. Alfred student is the student of Morgan. He showed that the position of the distance of gene on chromosome show the variation. If they are tightly packed, less variation, less recombinant. Loosely packed, large distance, they show higher recombinant. He proposed unit to measure the genes on chromosome. In eukaryote, this unit is known as centimorgan. The unit of distance measuring of gene on chromosome in eukaryote is centimorgan, whereas in prokaryote the unit is known as map unit. What is map unit and centimorgan? Centimorgan and map unit are the units by which we can measure the distance of genes on chromosome. In eukaryote, if we measure the genes in eukaryote on chromosome, then the unit is used centimorgan. If we use the distance measuring of gene on chromosome in prokaryote, the unit is map unit. So, this is linkage and recombinant. What is linkage? Physical association of gene on chromosome is known as linkage. Recombinant, when we crossing two parental with each other, the progeny is non-parental, means the progeny which is not resembled to the parent. That is known as recombinant. This recombinant and physical association gene represent the different ratio after the dihybrid crossing, not according to the Mendel's law of independent assortment. Clear?